Hi everyone, I'm Jasmine Satore, and in this video, I'm going to tell you guys a bit information about the interesting ingredients of our lives called genomes, and we will see what it is made of and why is it so much important for us. Also, we will see what are the wonders can be done with genomics. So without further ado, let's start. Do you know what is the one thing that make us us? What determines our traits and characters? In every organisms, whether it's a virus, a bacteria, a plant, animals, or humans, genome is present in all. Genome is the entire set of genetic material in an organism. In eukaryotes, it includes the nuclear genome that has DNA, which is appropriately condensed to form chromosomes. Then there are circular mitochondrial genome in animals, and plants have additional plasterial genome. The totality of all these genetic materials is termed as genome. In prokaryotes, the genome is smaller and exists in the cytoplasm of the cell. Prokaryotes may have additional genomes. For example, the plasmids organisms like viruses can have RNA as a genome. Genome is made up of DNA or deoxyribonucleic acid, or it may be RNA or ribonucleic acid. Genome is made up of genes and intergenic regions. The genes made RNA that code for proteins or non-coding RNAs, such as rRNA, tdRNA, microRNA, long non-coding RNAs, pyrRNAs, and etc. Both the coding and the non-coding RNA may have structural, functional, and regulatory role. On the other hand, the intergenic regions were largely considered as junk DNA because their significance in the genome was not known. But newer findings suggest they have include promoter enhancer regions and other regulatory elements. Our DNA or genome is nothing but biologically informations written with 4 chemical bases or nucleotypes. These are more commonly seen as a compact and organized structure with proteins called as chromosomes. But why do we have a genome? To me, the genome serves 2 major roles. First, just like our hard drives or SSDs, which stores important informations in digital forms of 0 and 1s. The genome stably stores the biological informations in the forms of a chemical bases, which can be replicated and transferred from one cell to another identical cell through mitosis, or it may recombine during meiosis, giving rise to somewhat different organisms, generating more diversity. The next important thing is that information stored in the genome can be developed or expressed in a specific manner, which means although all our cells have nearly identical DNA, the expression of one or more genes occur in a regulated manner. The gene expression is altered by many factors: our environment, our thoughts, our feelings, the food we eat, our age, etc. The end product of the gene expression is a phenotype. How an organism looks, its immunity against diseases, yield of a crop, its nutritional characteristics, and others. The such factors affects our DNA, making certain genes to express producing a particular phenotype that makes us, and determine our attributes. Genomics is the branch of genetics in which we study the genome. What it is made of, the sequence of the genome. We study the genome structure or its organization, number of genes, their locations, intron and exons, regulatory elements, repeats, and others. In functional genomics, we use different approaches like tDMA insertions, EMS mutagenesis, genetic mapping, gene expression analysis, and etc. to determine its functional significance. In comparative genomics, we compare the genome sequence and organizations of various organisms or species to see the genetic relatedness diversity, or evolutionary relationship among them. Coming on to what wonder we can do with the informations of the genome. Once we have the informations about the genome, we can find which genes or genomic regions are responsible for a particular trait of an organism. And once we know that, it's the role of scientists to play around with those one or more genes in a manner to develop better variety, or cure of disease, or produce a drug. Using breeding, mutagenesis, genetic engineering, and other approaches, you can recombine traits, knock out or stop a gene from expressing, or you can suppress expression of a gene or overexpress to get higher copies of a protein. You can also make an organism to express a newer protein which was not there earlier. You can engineer a protein with better properties and functions. So these are some of the major wonders that can be done with the genomic informations. It's the genome that make us us. And with the genomics, we explore the biological code to serve for better. So that's all for today's video. Again, I'm Jasmine Satore, and I hope that you learned a lot from my report about genomics. So thank you and goodbye.